Hello everyone. In this class, we'll learn about audition. Structure of the ear. The ear consists of three parts, namely external ear, middle ear, and internal ear. External ear is formed of two parts, that is auricle or pinna and external auditory meatus. The external auditory meatus uh, is of 24 millimeters in length. External ear funnels sound waves to the external auditory meatus. From the meatus, the external auditory canal passes inwards to the tympanic membrane. So this is showing structure of the ear, which is made up of three important parts: external, middle, and inner ear. External ear, it is made up of pinna and external auditory meatus. The middle ear, it is mainly between the tympanic membrane and the oval and round window, and it has the bony ossicles malleus, incus, and stapes. And the inner ear mainly consists of the cochlea, so which has the plenty of hair cells. Middle ear. The middle ear is an air-filled cavity in the temporal bone that opens via auditory tube into nasopharynx and through it to the exterior. The three auditory ossicles, malleus, incus, and stapes, are located in the middle ear. The ear ossicles are arranged in the form of chain extending across the middle ear from tympanic membrane to oval window. The function of these ear ossicles is to magnify the intensity of sound waves to about 1.2 to 1.3 times. Functions of middle ear. Tympanic membrane acts as a resonator. That is, it starts vibrating in and out and freely when the sound waves strikes the tympanic membrane. Another important function of middle ear is impedance matching. Sound waves have to pass from air into liquid medium in the internal ear. So there will be resistance to the transmission of the sound waves. This is overcome by increasing the pressure of sound waves at the level of oval window. The mechanisms are: the ear ossicles moves as a single unit. The size of handle of malleus is less than that of the size of incus, while the size of stapes is less than that of incus. And moreover, the handle of the malleus is longer than the long process of incus. Auditory ossicles thus function as a lever system that converts the resonant vibrations of tympanic membrane into the movements of the stapes against the perilymph-filled scala vestibuli of the cochlea. The lever action of the malleus and incus multiplies the intensity of sound waves to about 1.3 times. Then the effective surface area of tympanic membrane is 50 to 54 millimeter square, and that of oval window is just 3 to 4 millimeter square. That is, there is a reduction in surface area by 17 times at the level of oval window. Thus, both these mechanisms lead to corresponding increase in the sound pressure at the oval window. So the total increase in the sound pressure is about 22 times, that is 17 into 1.3. Tympanic or acoustic or attenuation reflex. It is a protective reflex against loud sound. Loud sound initiates a reflex contraction of middle ear muscles. First, it causes the contraction of the tensor tympani, which pulls the handle of the malleus medially, which makes the tympanic membrane more taut and decreases the vibration of tympanic membrane. The tensor tympani is the muscle attached to the neck of the malleus, and it is supplied by the fifth, that is trigeminal nerve, by mandibular division. And second is stapedius is attached to the neck of stapes, which is supplied by the seventh or facial nerve. Its contraction pulls the foot plate of stapes away from the oval window and thus preventing the transmission of sound waves to the internal ear. Both these muscles contract reflexly in response to loud sound, and thus excessive sound is prevented from going to the oval window, and this reflex is called as tympanic, acoustic, or attenuation reflex. The middle ear also helps in equalization of pressure on the either side of the tympanic membrane. It equalizes the pressure on both the sides of the tympanic membrane with the help of eustachian tube. Eustachian tube is usually closed but during swallowing, chewing, and yawning, it opens and equalizes the pressure on both the sides of tympanic membrane. Coming to the inner ear, that is also called as labyrinth, is made up of two parts, the bony labyrinth and membranous labyrinth. The bony labyrinth is a series of channels in petrous portion of temporal bone. The membranous labyrinth is present inside these channels and it is surrounded by fluid called perilymph. This membranous labyrinth more or less duplicates the shape of the bony channels and it is filled with a fluid called as endolymph. There is no communication between the spaces filled with endolymph and those filled with perilymph. So this diagram shows the middle ear and the inner ear. When the sound waves falls on the tympanic membrane, the ossicles they vibrate and the vibration is transmitted to the inner ear through the to the cochlea, and cochlea has the three parts: scala vestibuli, scala media, and scala tympani. Scala vestibuli continues with the scala tympani at the helicotrema. So whenever sound waves falls on the oval window, the sound waves produces vibration in the scala vestibuli, so which comes back through scala tympani. Okay, so vibration of these scala vestibuli, it also leads to vibration of the scala media, so which harbors the organ of Corti. Within there'll be hair cells. The distortion of the hair cells is going to produce the action potential which reaches the brain, and the sound is sensed by the brain in this way. The internal ear has divided into three chambers, that is the scala vestibuli, scala media, and scala tympani by two membranes, that is racemous membrane and bacillus membrane. Scala vestibuli and scala tympani are filled with perilymph and are continuous at the apex, that is through the helicotrema. Scala media is continuous with membranous labyrinth and it is filled with endolymph. Here you can see the cross section of the cochlea, so which through the two membranes, that is basilar membrane and racemous membrane, divides the canal into the three parts: scala vestibuli, scala media, and scala tympani. Scala vestibuli and scala tympani, they are continuous and they have in them the perilymph, whereas scala media it is filled with the endolymph. And here you can see the organ of Corti, so which is a sense organ of audition, which harbors many hair cells in them. Organ of Corti. It is located on the basilar membrane and it contains hair cells. Hair cells are the receptor cells that transduce sound energy into electrical energy. It extends from the apex of the base of the cochlea and has a spiral shape. There is one organ of Corti in each ear. The process of hair cells, the PS, the tough membrane like reticular lamina that is supported by rods of Corti. The hair cells are arranged in 4 rows and 3 rows of outer hair cells lateral to the tunnel formed by the rods of Corti and 1 row of inner hair cells medial to the tunnel. There are around 3500 inner hair cells which are important for fine sensory transduction, and there are about 20,000 outer hair cells which is responsible for detecting the presence of sound. Outer hair cells are capped with gelatinous material called as tectorial membrane. Cell bodies of afferent neurons that surround the bases of hair cells are located in the spiral ganglion. About 90 to 95% of fibers innervate inner cells and only 5 to 10% innervate outer hair cells. In each hair cells, there are about 30 to 50 hairs called stercocelia, and it has actin and myosin. Very fine processes called tip links tie the tip of each stercocelia to the side of the its higher neighbor. At the junction, there are cation channels which are mechanically sensitive. When hair cells move in one direction towards the longer stercocilia, channels open up and there is the influx of potassium and the calcium. If the cells move in opposite direction, the ion channel closes. The supporting cells are in inner phalangeal cells and outer phalangeal cells. Inner phalangeal cells, they support the inner hair cells, and outer phalangeal, they are also called as diater cells, support the outer hair cells. The outer phalangeal cells are supported by Hansen cells, which again supported by the Claudius cells. The junctions between hair cells and other supporting cells are tight junctions to prevent mixing of the endolymph and perilymph. So this is the structure of organ of Corti, so which is based on the basilar membrane, and it is made up of a tunnel and medial to which we have the inner hair cells and lateral to which we have outer hair cells. Inner hair cells are less numerous compared to the outer hair cells, and the tips of the hair cells, they are embedded in the tectorial membrane, mainly that of the outer hair cells. And you can see the nerves which forms the spiral ganglion from hair cells, so which helps in the transmission of the action potential from these hair cells. And the other supporting cells for the outer hair cells are cells of Claudius. Sound transduction. Sound waves which enter the cochlea gets transmitted along the scala vestibuli and spreads towards helicotrema. At the same time, sound waves are also transmitted to the fluid into other chambers. This will lead to the vertical up and down movement of basilar membrane. This results in up and down and side to side movement of hair cells. The process of hair cells project into the endolymph, whereas bases are bathed in perilymph. The mechanism of production of action potential. When tip links pulls the stercocilia, the ion channel opens and there is a potassium influx because endolymph consists of more amount of potassium. The receptor potential occurs in the hair cells, which causes the calcium influx, which causes the depolarization of the hair cells in the lower part of the hair cells, and that causes the release of neurotransmitter, that is glutamate, into the synaptic cleft between receptor cell and afferent nerve, sensory nerve. And neurotransmitter eventually binds with a specific receptor on the sensory nerve fibers, and ligand gated ion channels open, which causes action potential in the cochlear division of the 8th cranial nerve. And then the transmission is going to reach to the brain through the auditory pathway. Auditory pathway. The afferent fibers in the auditory division of 8th cranial nerve is the first order neuron, and it ends in the dorsal and ventral cochlear nucleus. So there are 3 sets of fibers from this level, that is the second order neurons. The first is dorsal cochlear nucleus. The second order fibers cross to the opposite side from here and ascend and end in inferior colliculus. There will be many collaterals given to the reticular formation. From here, third order neurons arise and they end in the medial geniculate body of the same and also of the opposite side. From here, fourth order neurons arise, they end in the auditory cortex, that is primary auditory area 41, association area 20, 21 and 22. Some fibers from the ventral cochlear nucleus, which forms the second order neurons, cross to the opposite side, they end in the trapezoid body. From here, third order neuron arises, they end in the inferior colliculus, and the fourth order neurons arises from the inferior colliculus, they go to the medial geniculate body, so there won't be any relay to end, and ultimately they end in the auditory cortex. Then from some fibers from the ventral cochlear nucleus, which forms the second order neurons, end in the superior olivary nucleus. From here, third order neuron arises, they end in the medial geniculate body, which also gives collaterals to the inferior colliculus and reticular formation. From here, fourth order neuron arises, they end in the auditory cortex. So this is the diagram showing the auditory pathway which are going to form mainly the dorsal cochlear nuclei and ventral cochlear nuclei. So dorsal cochlear nuclei, the second order neuron arises from here, they goes to the superior colliculi, and from superior colliculi it is going to go to the both the sides of medial geniculate bodies. From there, fourth order neuron goes to the cortex. If you see the ventral cochlear nucleus, some of the fibers they goes to the trapezoid body, and from the trapezoid body the third order neuron arises. They are going to go to the lateral geniculate body, medial geniculate body. From there they go uh, to the auditory cortex, and some of the fibers from the ventral cochlear nucleus they go to the olivary superior olivary nucleus of the same side, then they are going to ascend in the same side to end in superior colliculus. From there the information goes to the auditory cortex. Efferent pathway. Some fibers from cortex descend and ends in outer hair cells. Neurotransmitter released here is acetylcholine. Although these inhibitory fibers end in outer hair cells, they actually modify the activity of the inner hair cells. Salient features of the pathway are there is a bilateral representation due to the extensive crossing of the fibers, and there is also descending pathway, and there occurs integration of visual and auditory impulses due to the connection between superior and inferior colliculus, and general arousal also is possible with this pathway as it gives collaterals to the reticular formation. Some of the theories of hearing are traveling wave theory or place theory. The movement of foot place of steps sets up a series of traveling waves into the perilymph of scala vestibuli. Basilar membrane is the mechanical analyzer of the sound frequency. The basic pattern of movement of basilar membrane is like a traveling wave. High frequency sound waves produce maximum height at the oval window, and low frequency sound has a maximum height near the apex. Correspondingly, the basilar membrane near the oval window vibrates in response to high frequency sound, and the basilar membrane near apex vibrates in response to low frequency. So that is different hair cells responds to different frequencies of sound depending on their location on the basilar membrane. So this is applicable to frequencies between 2000 to 20,000 hertz. So thus human ear can discriminate the sound of frequency between 20 to 20,000 hertz only. Wallace principle theory. Theory of Wallace principle is applicable for low frequency of sound waves which are having less than 2000 hertz. There is a relation between frequency of sound and action potential developed in the sensory nerve endings. For low frequency sound, frequency of action potential is less, and if greater frequency, a volley of action potential is produced, that is, more number of action potentials are produced. Masking. It is a phenomenon to produce inaudibility of one sound by the presentation of another. In audiometry, one ear is kept busy while other being tested by sound. So this diagram shows the Wally principle that the base is going to detect the higher frequency sound waves, and whereas apex is going to detect the low frequency sound waves. That is, the hair cells at around the base they are sensitive to only high frequency sound waves, whereas hair cells which are present in the apex they are sensitive to the low frequency sound waves. Deafness. It is inability of an individual to hear either wholly or partially. The deafness is of two types: conductive deafness and nerve deafness. Conductive deafness, here the defect is in the conduction of the sound due to the impaired sound transmission. Some of the causes for conductive deafness are ear wax, defect in vibration of tympanic membrane, infection of the middle ear, that is otitis media, otosclerosis, or eustachian tube blocked due to the common cold. Nerve deafness occurs because of the damage to the hair cells in the neural pathway. Some of the causes could be hereditary, injury to the eighth cranial nerve, drugs like streptomycin, vascular causes, or maybe industrial noise pollution. Tests for hearing, they are mainly the watch test, tuning fork test, and audiometry test. Auditory acuity, that is sharpness of hearing, can be measured with the help of an audiometer. This test helps to assess the degree of deafness and the frequency range in which deafness is most affected. The device consists of the following parts, that is electronic oscillator, intensity dial, and headphone. These are some of the common tuning fork tests which are done: Rene's test, Weber's test, and Schauberg's test. In Rene's test, the base of the vibrating tuning fork is placed on the mastoid process until subject no longer hears it. That is bone conduction, and then it is held in the air next to the ear, which is going to test the air conduction. Normally, subject hears vibration in air after bone conduction is over. Air conduction is better than bone conduction. That is Rene's test positive. In conductive deafness, vibration in air not heard after bone conduction is over. That is, bone conduction is better than air conduction, where the Rene's test becomes negative. In nerve deafness, if partial nerve deafness, Rene's test is positive. If complete nerve deafness, both air conducted and bone conducted sounds are not perceived. In Weber's test, base of the vibrating tuning fork is placed on the vertex of the skull in the middle or over the mandible. Normally, both the ears hear the sound equally well. In conductive deafness, sound is better heard in the affected ear, whereas in nerve deafness, the deaf ear remains deaf due to the bone conducted sound, and sound is thus better heard and perceived by the normal ear. That is, it is lateralized to the healthy ear. Schauberg's test is done by bone conduction. It is going to determine the bone conduction of the subject compared with that of examiners, assuming that the latter is normal. So here, in normally, both the subject and examiners hear the sound waves equally, and in conductive deafness, subject's bone conduction is better than that of examiner, and in nerve deafness, subject's bone conduction is worse than that of examiner. Vestibulo-ocular reflex. It is actually a reflex that maintains visual fixation on the stationary points while the body rotates. When rotation starts, the eyes move slowly in a direction opposite to the direction of rotation, maintaining visual fixation. Nystagmus, the regular and rhythmic to and fro involuntary oscillatory movements of the eyes. Meniere's disease, or it is also called as endolymphatic hydrops, is a disorder of the inner ear where endolymphatic system is distended with endolymph. And endocochlear or endolymphatic potentials are potentials and a potential difference about plus 80 millivolts exist in all the time between endolymph and perilymph, with the positivity inside the scala media and negativity outside. And it is generated by the continual secretion of potassium ions into the scala media by the stria vascularis. So this is in brief about the audition. Thank you.